Okay, in the last video we went over an introduction to gluconeogenesis. In this video, we're going to start examining the various reactions that are done to bypass those three irreversible reactions from glycolysis. Because for the most part, we can take advantage of the fact that all the other glycolytic reactions are reversible. Okay, so the first reaction that we need to deal with is the synthesis of PEP. Okay, so typically this starts with pyruvate. Okay, other substrates can be converted through intermediate enzymes and things of that nature to get to pyruvate. Okay, and we're going to acylate this or carboxylate this. Okay, depending on how you want to think about it. Okay, this is mediated by the enzyme pyruvate carboxylase. Okay, and it uses biotin as a coenzyme. Okay, and you all know what a coenzyme is. This will consume 1 mole of ATP per mole reacted, and it will eject 1 mole of ADP and 1 inorganic phosphate per mole reacted. Okay, what we get is we get a molecule called oxaloacetate. Okay, which looks like this. And we get some remaining acid. Okay, this is oxaloacetate. Okay, and for convenience sake, it's often abbreviated OAA. Okay. The structure of biotin should be on page 461 of your text, so I would encourage you to look that up. Biotin is a very important coenzyme, not only for this process, but other processes in your body. And it's really important in bioanalytical chemistry because we can use a bacterial protein called streptavidin. And streptavidin will bind to biotin with an affinity that approaches the strength of a proper covalent bond. So it's used very frequently for click sensors, chemical click sensors in bioanalytical chemistry. Random aside, I know, I apologize. Okay, this happens in the mitochondria. Okay, so this happens in the cell mitochondria. Largely. Okay, largely. In some species, it's found, it's done exclusively in the cell cytoplasm. Okay, for humans, it's done in both places, both in the cell cytoplasm and in the mitochondria. We have a problem, though, in that oxaloacetate is not permeable to the mitochondrial membranes. So, in order for it to be transported out of the mitochondria and used further, it will sometimes have to go through the Malate Shuttle. Okay, and the Malate Shuttle is a reversible reaction. Okay, that is mediated by malate dehydrogenase. Okay, it is a reversible reaction that consumes. Okay, it consumes NADH. Sorry, that should be a capital. And H plus. Okay, and what it does is it converts the oxaloacetate to malate. Okay. All right. So in organisms that cannot process the oxaloacetate further in their mitochondria, or in cells where that mitochondria is not capable of doing so, we convert it to malate. Okay. We convert it to malate, consuming one NADH and a free acid to produce NAD plus and this malate. And this can be transported across the mitochondrial membrane. And once in the cytoplasm, it's converted back into oxaloacetate. Okay? Because oxaloacetate is used in the next reaction. This can occur either in the mitochondrion or in the cell to produce our PEP, right? So we take our oxaloacetate, right? And the enzyme PEP carboxykinase converts that into PEP, okay, with a mole of CO2, okay? And this conversion requires GTP, okay, which is the guanine analog of ATP, and it produces GDP, okay? So if you recall back when we were talking about energetics in biochemical systems, right, we said ATP is mostly the primary fuel of the cell, with NADH being its primary electron transport mechanism. Well, GTP is its secondary energy transport molecule, okay? It functions very similarly to ATP, okay? But some enzymes cannot interact with ATP. They need the different nucleobase, okay? All right. As I said, for humans, okay, this PEP synthesis happens both inside and outside our cell mitochondria. So sometimes we will have some mitochondria will process the whole thing, and it'll emit PEP. PEP is able to get through the mitochondrial membrane, no problem. Sometimes the mitochondrion cannot process it beyond oxaloacetate, and so then we have to convert it to malate through the Malate Shuttle to get it out of the mitochondrion and into our cell cytoplasm, where this reaction then takes place, okay? But that gets us part of the way there. Once we have PEP, we can then take it all the way back to fructose 1,6-bisphosphate using the reversible reactions for glycolysis, okay? Once we get to fructose 1,6-bisphosphate, though, we have another barrier to overcome, okay? So we have to convert fructose 1,6-bisphosphate to fructose 6-phosphate, right? We have to remove that singular extra phosphate group, right? We have to hydrolyze that, okay? And so that has a reaction, right, that looks like this. And again, I'm using, the shorthand. I'm not going to draw out the various phosphate groups. I don't think that's necessary. I think you all know what a phosphate group looks like, right? So we have our fructose 1,6-bisphosphate, right? There we go, right? We add to that water because this is, a hydrolysis reaction. Right? Now, guess the enzyme. Yes, it feels like Jeopardy. Okay, time's up because I don't want to have the video get copyright struck or something. Okay, so fructose, 1,6 bisphosphatase. I don't know why it disappeared and reappeared. I don't know why things I write do that. Not a clue. Try to ignore it, okay? You'll notice I'm writing this as an irreversible reaction, and that's because the delta G, right, the change in the Gibbs free energy for this, okay, is negative 16.7 kilojoules per mole. Okay, and you might think, oh, well, that means this is somehow giving us energy. No, it's not. It's not. The overall pathway still consumes energy, okay? And we'll get to that in a minute. All right. So what we get out of this, okay, is we get our monophosphate fructose. And again, even though I'm not drawing the bold lines, these are the Haworth projections. Okay. Okay. Plus, we get an inorganic phosphate. Okay. Under our cellular conditions, we have this distinct exergonic reaction that's irreversible. Okay. The enzyme here is allosterically inhibited. Okay. So that enzyme is allosterically inhibited. Okay. By its own starting material, ironically. Okay. By fructose 1,6 bisphosphate and AMP. Okay. It's accelerated by the presence of citrate. Okay. So once we have fructose 1,6 fructose 6 bis. Oh my goodness. I am just insisting on trying to say that once we have this. Once we have fructose 6 phosphate, right, we can then use reversible reactions from the glycolytic pathway to get us to glucose 6 phosphate, right? So we can have G6P, right? And that will be converted to glucose, right? We dephosphorylate, right? And this happens exclusively in the liver, okay, by glucose 6 phosphatase. Okay. This happens exclusively in the liver. Okay. Glucose 6 phosphatase is only present in the liver. All right. So this final stage has to be done by your liver every time. Every time. Okay. And we get our inorganic phosphate out. Okay. Sorry. Liver, kidney, and intestines. This is what happens when I don't read my notes. Okay? So liver, kidney, and intestines. Okay? There are no other organ systems that possess this enzyme. Okay? Not anywhere else in your body can this take place. Only these 3 organ systems. Period. Okay? In fact, not even all the cells in these organs can handle this reaction. There are specific cells in each tissue that have this enzyme, and they are the only cells in which this enzyme is found. Okay? So you can imagine how much strain you're putting on those particular cells if you're saying, yeah, I'm just going to stop eating carbs. Right? Not a good idea. Okay? Energetically speaking, we consume 6 phosphate bonds. Per mole of glucose. Okay? So to produce 1 mole of glucose, we consume 6 phosphates. All right? Now, what I would like for you to do is to figure out how much energy this costs us. Okay? Per mole of glucose produced. Okay? So you know the mole ratio is 6 phosphates to 1 glucose. Figure out what the energy is for 1 phosphate bond, that way, hydrolysis. That way you can figure out how much energy is consumed to produce 1 mole of glucose in your blood or in your body. All right? And I want you to put that in the comments below this video. Okay? And I will be looking. So I want to see all 4 of you comment on this. All right? Because I want you to understand how energetically costly it is for your body to do this. Okay? And I want you to understand that that energy has to come from somewhere. Right? So I want you to not only compute how much energy is consumed to produce a mole of glucose in your body, but I want you to calculate how much energy is produced by burning a mole of glucose in your body, right? And I want you to make note of any disparity you see between those two, right? Do we get more energy than we put out? Or should I say, do we get more energy out of the glycolytic pathway than we consume? Are they completely or nearly completely reversible? Are they about equal? Think about that. I want you to think about that. I want you to discuss it. Okay? And then I want you to post a comment on this video. All right? I hope you all had an excellent break. And I'm looking forward to moving on with our studies here of metabolic pathways and getting into nucleic acids. I'm really looking forward to nucleic acids and protein, synthesis and translation and all that stuff. So it'll be a good time. All right. I'll see you in the next video.